It's a great pleasure for me to introduce our guest speaker today, Poggio from MIT. Tommy's career is incredible in terms of scope and achievements, so I will not try to review it, but what I'll do instead is I'll read to you the description of Tommy as it appears on the MIT website, and I think you'll get the idea. Tomaso Poggio is one of the founders of computational neuroscience. He pioneered models of the fly's visual system and of human stereo vision, introduced regularization theory to computational vision, made key contributions to the biophysics of computation and to learning theory, and developed an influential model for cognition in the visual cortex. Tomaso Poggio is now a Eugene McDermott Professor at the Department of Brain and Cognitive Science at MIT, co-director for biological and computational learning, member for the last 27 years of the Computer Science and Artificial Intelligence Laboratory at MIT, and since 2000, member of the faculty of the McGovern Institute for Brain Research. So, born in Genoa, in Italy, in 1947, he received his doctorate in theoretical physics from the University of Genoa in 1971, and then became a research assistant in the Max Planck Institute for Biological Cybernetics in Tübingen, Germany, from 1972 until 1981, when he became an associate professor at MIT. He is an honorary member of the Neuroscience Research Program, a member of the American Academy of Arts and Sciences, and a founding fellow of the AAAI. I didn't know that. He received several awards, such as the Max Planck Research Award, the MIT 50K Entrepreneurship Competition Award, the 2003 Gabor Award, and the 2009 Okawa Prize, and there are a few more. He's one of the most cited computational neuroscientists with an age index greater than 90 based on Google Scholar. So that's the facts, and I think they speak for themselves. On a personal note, I spent 4 years in Tommy's lab as a postdoctoral fellow from 1987 to 1991, and it was 4 of the best years in my life. I would have stayed there longer if they let me. And there are two reasons. Of course, the scientific excellence of MIT was one reason. That's to be expected in MIT. But I think the main reason that it was so pleasant and so wonderful is the atmosphere in the lab, the supportive atmosphere that's due to Tommy's generous and kind nature. So I'm very happy to introduce Tommy. And I think before I have to be giving you a signed.
Oh, thank you. Okay. Fantastic. Thank you very much to the center and Chaim, Idan, and to Daphna for the wonderful introduction. By the way, I wanted to keep her, but it's great to be here in front of so many friends and collaborators, and thanks to everybody for coming. I want to tell you today a simple message in my talk. And the message is something that probably people here at ICNC know, but that I realized really just not many years ago, like 3 or 4 or so, that neuroscience, computational neuroscience, may be just at the point in which it begins to provide useful ideas, new ideas and approaches to machine learning, artificial intelligence, maybe even computer science over the next few years. And so in order to tell you how, at least I personally came to this belief, to this bet, let me tell you what we have been doing recently for the last few years in my group. We have been working for a long time, 20 years or so, on the problem of learning, machine learning, learning from examples, how to make computers become more intelligent from experience. Thank you. From experience rather than from programming. And the problem of learning has at least 3 different levels of analysis: mathematics, computer algorithms, implementations, and also the related problem of how the brain learns. And these 3 areas, 3 directions of research is something we have been following for the last, as I said, 20 years or so, from the level of theorems on the foundations of learning to algorithms that one can use in engineering application for in the area of bioinformatics, of computer visions, of computer graphics, of finance, and so on. And also, as I said, the question of learning as the related question of how the brain learns, or how visual cortex learns to recognize objects as a particular case in point. In the past, the connections were in some cases a bit virtual. So there was not much that neuroscience was telling to engineering or mathematics. And in fact, for instance, at the times in which Daphna was in my group, the research in computer vision was kind of separate from the research on the neuroscience of vision. But 4 or 5 years ago, it turned out that a model that was kind of a relatively stupid model, trying to mimic what we knew about parts of the visual cortex, part of the cortex, this model did work also pretty well when compared to engineered computer vision. And so that was an interesting surprise for me, and the realization that these errors could become much more real than in the past. And just to give you a feeling, this is the background. So before this kind of realization, my group and several other people worldwide started to work on the problem of finding faces in objects quite some time ago, 15 years ago or so. And these are some of the results of the work at the time. In the meantime, of course, you can buy almost any digital camera, and it will have a face detector included in it, and it will work probably better than, this was pretty good. On the other hand, this is an interesting case of a false alarm. Anyway, it takes quite some time to go from research to the market. And for instance, this is happening on another piece of work that we did also long ago. This was work triggered by collaboration with Daimler at the time for detecting pedestrians. In the meantime, there is a great company started by a faculty member here, Amnon, somewhere, with commercializing not the same technology, but evolutions of this initial laboratory work. And so this was, as I said, work in pure computer vision, not really informed by the brain. But then, as I said, we got interested in the brain, and we were also interested in overcoming some of the limitations of the techniques I've shown you before, which did work well, but certainly not as well as humans. And so we started looking at visual cortex, and one of the first things you have to recognize is that visual cortex has an organization which is quite different from the learning algorithms that we're using, like support vector machines or similar classifier. But before getting into this, for the ones of you who are not expert in computer vision or vision, I want to make the point that the problem I'm dealing with here is the problem of recognizing or detecting the presence of an object, say an animal, for instance, or it could be a car, in an image. And the problem is surprisingly difficult. Surprisingly difficult if you don't constrain in any way the kind of images that you're dealing with. This is the problem in a sense, is the main problem of vision, to find what is in the image. David Marr, who was a friend, a collaborator of mine, defined vision in his book as the vision is answering the question of what is where. And this was, he wrote a book at the time in which we together worked at the Salk Institute visiting Francis Crick. This was in 76, I think, or 78. And anyway, so what is where? I will speak today mostly about the what part of the problem. What is in the image? Find whether there is, for instance, an animal or not in the image. Now, just to give you again, to the non-neuroscience expert, some numbers. The human brain, nobody knows for sure, but it's around 10 to the 11 neurons. This is about 1 million flies. It's a quick mnemonic to remember. The fly is about 300,000 neurons or so. And it has a much larger number of connections that typical electronic components have. So there are about thousands or more synapses or contacts between cells than, whereas in a typical component on a chip has probably in the order of 3 connections or so. The part of visual cortex that is involved, as people think, in the what problem, what is in the object, that part of visual cortex has probably around 1 billion neurons, which is a big number, but is not really big in this era of terabytes and petabytes and exabytes. So that's just to give you the background, and the ventral stream itself consists of a series of visual areas that go from the back of the head here, the eyes here in the front, from the back of the head to the front, up to areas which are not visual anymore, prefrontal cortex, where decisions are taken. And, and here, what physiologists know, trying to summarize in one slide, 4 decades of work, what physiology know about it is, first of all, originated with the work of Huber and Wiesel, who got the Nobel Prize for this 3 decades ago. And these are cells, properties of cells in V1, or primary visual cortex, which are divided in 2 main classes, and there are probably many more. And the 2 classes are so-called simple cells and complex cells. And the simple cells will be activated if you present, maximally activate one cell by a bar of a certain orientation, typical for this cell, at a certain position in the visual field. So they are kind of bar detectors, although they are not really bar detectors, they're more like filters. And there are cells responsive to different orientations, and cells responding to the same orientation but in different position of the visual field. And then there are complex cells that are also optimally responding to a bar of a certain orientation, but are more invariant or tolerant to the exact position of this bar within a certain area, which is called a receptive field of this cell, the area of the visual field in which you can activate this cell, the electrical activity of this neuron. Now, as you go from V1 to higher areas, like V2 and V4 and posterior IT and anterior IT, you get cells that seems to be maximally stimulated by more and more complex patterns, and up to the point that in anterior IT you may have some cells responding to drawings of a face, and in some cases possibly to individual faces, with a much higher degree of selectivity than in earlier areas. At the same time, the receptive field, the part of the image that the cell sees, is stimulated from, become bigger and bigger as you go higher up. And it seems also that there is more and more tolerance to the exact position and size of the optimal stimulus. In other words, for instance, this drawing may stimulate a particular cell, irrespective of whether it's here or displaced by, say, a couple of degrees, or double in size. So you have this increase in specificity, increase in tolerance as you go from V1 to anterior IT. Now, to give you an idea of the times taken by the wave of information going from the retina through this series of modules, here are data collected from different papers from Simon Thorpe. And so it starts with the retina. It takes in the order of 20, 40 milliseconds. The longer times are for humans. The shorter time are for monkeys. Going from the retina to the LGN, and then to primary visual cortex, and then up to anterior IT, 80 milliseconds or so, and then up to regions that are really involved in taking decisions. So they're not visual, purely visual anymore. And these are, again, these areas, V1, V2, V4, and IT. Now, it's a reasonable hypothesis that this hierarchy of processing stages plays a role in our visual ability, now how good we are in recognizing objects and processing images, in perceiving. So part of the puzzle is how is this done and why hierarchies are important. I'll speak more on Thursday at another talk here about the fact that standard learning algorithms essentially consist of a very shallow architecture in which you have only one layer between inputs and outputs. And here we have many layers, looks like, between the input and the output. So it's an interesting question, why and how is that exploited? Now, I'm speaking today mostly about the feed-forward connectivity, going from lower areas up to higher areas, and I'm neglecting for today, or at least until the discussion, the role of the back projections. For every forward projection, there are also back projection, feedback projection, if you want, from a higher to a lower area. The model we had started to develop a long time ago, my goodness, almost 10 years ago, is essentially one of a family of models. I'll give you a number of citations in a second, but it's a feed-forward model of hierarchy of layers of cells. You can think of them as neurons, which are static neurons. They output a number. I'll tell you about more of the biophysics that we think is possible, but for this model, think of them as simple processing units which are really doing only two basic operations. The first one is a tuning to an optimal stimulus. You can think of it as, for instance, a Gaussian, in this case, centered on a horizontal bar. So the best activation would be a stimulus of the same type. You can think of this also a scalar dot product between a template, this horizontal bar, and the input consisting of an array of inputs from, say, ganglion cells in the retina, or if you want, of pixels in the image. And so the most similar input to a bar would be the one that will stimulate these cells most. So, and then the next stage would be a max operation, or a max-like operation, on a set of inputs to a complex cells. And the set of inputs to a complex cells would be from simple cells in different positions, but with the same selectivity. For instance, this oblique bar, and different scales for this oblique bar. So these are the two operations which are repeated alternatively in each layer. So simple for a Gaussian tuning and complex for this max-like operation. And at the next stage, as I'll describe later more, the tuning will be, instead than on a bar, will be on essentially conjunction or composition of bars, of these filters, but the composition is learned in an unsupervised way, and I'll tell you more about this later. And then, after iterating this basic structure, you get to a point in which a classifier, you can think of it depending on, could be a linear, it is usually a linear classifier, is looking at this transformed image, if you want. This would be at the level of IT, and the classifier itself could be circuits of neuron in, say, prefrontal cortex of some structure of that type, and this would be a classifier for answering a what question, for instance, is or is not a, say, a car in the image. So that's the basic architecture. As I said, this is just a perhaps more faithful to the physiology and the anatomy version of a class of models that have been described originally, in a sense, by Hubert Wiesel when they had their simple, complex, and some rather vague notion of hypercomplex cells. But mostly by Fukushima, we had a very similar model, and there were other people, including Shimon Ullman, who came to it more from a computational perspective, and quite a few other people suggesting model of this general architecture. Ours, as I said, is just perhaps a bit more careful in terms of taking numbers, connectivity, numbers of cells, physiological properties of the cells from what one can get, especially from the physiology and the anatomical literature. And on the biophysics side, for the biophysicist among you, it's possible to think of these two basic operations in terms of simple circuits of Hodgkin-Huxley type neurons with, for instance, inhibition of the silent or shunting type. And we have done simulations of circuits that can do a Gaussian tuning or a max-like operation, but we have never implemented the full model in terms of realistic synapses and spikes. So it's still open question whether it could work in terms of the times involved, for instance, and the timings involved. So you can also think of this tuning operation, the Gaussian-like operation, as an AND-like operation. That's a kind of metaphor, but essentially you are looking for complex patterns which are the conjunction of simpler patterns at the lower level. And you can think of, again, as a metaphor of this max-like operation. Does not need strictly to be a maximum. We know, empirically we found that something similar to a maximum works, a linear average almost works. This is kind of an OR, essentially ORing between simple cells with the same selectivity but in different position and scales. And so you have this alternation of AND stages with OR stages, and so you have essentially a conjunction of this junction represented in this layered architecture. So in terms of the learning, I don't want to spend too much time on this. Let me just say that we are doing something very simple and probably not very biological in terms of learning these conjunctions at this level of S2 and S2b and S3, which is essentially storing the activity that is elicited, say, in a cell here by some natural image presented down here, and doing this through essentially unsupervised learning through random sampling, doing this layer, say S2 and then S2b and then S3, layer by a layer, one could do something more similar to find clusters of the most commonly appearing features at this level in the images and represent those as tuning, optimal tuning of the neurons. And we can discuss more how this is exactly done. It does not seem to be very critical, and there is much more that we should or should be done there. And then the only supervised part of this system is this last stage, where we have a linear classifier, or could be a support vector classifier, nonlinear support vector classifier, where here we are using a relatively small number of supervised examples of the task. For instance, images where there are animals labeled with 1 and images without animals, if this is the object we want to detect, labeled as 0. And as I said, that also means that the unsupervised learning is a kind of learning you do once for all, like in development of this visual system, and then this is the task, the only part which is task dependent, which you change depending on which particular object class you want to detect. So most of the work really in the last 5 years or so was trying to collaborate with physiologists, monkey physiologists, to try to test and also to tune or change different parts of the model. So this blue experiments with Nikos Logothetis a long time ago, and data coming from Huber and Wiesel and others, these are the ones which really we used to tune some of the parameters in the model, in addition to other papers. The red ones are kind of prediction or consistency with already existing data. So there are, we found with Ilan Lampe, with Narada Watzman and David Förster, that some complex cells perform a max-like operation, and there are other data from cells in V4 and IT that are consistent with this kind of this family of models, and also some psychophysics. Now, I want just to briefly mention just a couple of these. One is experiments with Jim DiCarlo, in which Chao Hung and Gabriel Kreiman, in which we record from infratemporal cortex and created a pseudo population. Think of this as recording from 200 neurons from infratemporal cortex. And then you can train a classifier. Training with input is the activity elicited by certain images, and output for the classifier, for the training set of the classifier, is the label of that image that you know. And then you can use this trained classifier to read out the information from the neural activity. In other words, you record new neural activity, and the classifier will tell you which image was presented to the monkey. So this is an example of how well the readout worked. It works pretty well. I didn't make some mistake from time to time. But anyway, what I wanted to show you is that the readout from this population of neurons in IT reveals properties similar to the model. So these bars here show that the readout itself is position invariant to a large extent, because you can use the classifier trained with a neural activity elicited by images of a certain size, at a certain position, and it will also work very well for images presented of a different position and of different sizes. Okay? So this, by itself, forgetting about the model, this experiment tells you that the representation in IT is largely position and size invariant. When I say largely invariant, I don't mean invariant over a huge number of degrees or sizes, but it's around at least plus minus 2 degrees of translation and a factor 2 up and down in size. Now, if you do readout from the model, so essentially recording from neurons in the model, and using the same stimuli and the same procedure, you get very similar results. These are these other plots here. By the way, interrupt, please, if there is something.
So,
yeah.
I'm confused. This is reading out from a population of
yeah.
So isn't it straightforward that there is a translation invariance because the population covers a whole range of populations?
Well, the image is presented, the classifier has seen the activity only from for the central position, right? So,
only from groups that respond to this position.
Well, let me go back for a second. So, so the image down here during the training of the classifier is in a fixed central position and of a fixed size. So in itself it's not obvious that if you have a network and you train, say in this case, suppose this is the classifier you train, that then it will respond in the same way correctly when you shift the image. Now, the model itself is trying to do this, right? But it's not clear how well it could do it and whether the performance, the type of invariance is the same, would be at the same level as the brain. So that's one it's one data where the model agrees with the experiments. If you do, for instance, this readout from V1, say, you will find out that the classifier trained with the image in the central position will not work at all if you displace the image, or very, very badly. So here is another situation in which you see here a kind of caricature of something which is called rapid categorization. People can look at an image which is flashed very briefly and still often understand or see what is there. We don't need to move our eyes or saccade or fixate to recognize the main feature in an image. And this is called, as I said, rapid categorization. And we used it as a way to force the visual system of a subject, a human subject, to work in a feed-forward mode. I'll not tell you exactly the argument, but one can argue that there is too little time for back projections to play a role. And so we expected, perhaps under these circumstances, to give a chance to the model to see whether it could mimic human performance. And we found, and I was actually surprised, Tomaso, Oliver found, doing this experiment, that the model could in fact perform at the level of human subject in a task of finding whether there is or there is not an object in images. And images are images of this type, ranging from close-up to images in which the far body one, where there is a lot of clutter. I think the battery is giving up. But so, for instance, here you see that the overall performance of the model is similar to humans, is 80% correct, roughly. And just to give you an idea, the task is more like this. So it's actually in which there is a mask after the image, which is presented very briefly. So it's more difficult than the ones you have seen in the first square. But still, people can typically do pretty well. And we found out also that images that are difficult for humans also tend to be difficult for the model, and vice versa. This is an example which is easy for the model and easy for humans. So there is a good correlation image by image. So, you know, this does not at all say that the model is correct, but that it may capture some aspects of this visual processing, perhaps. I want to mention briefly a couple of, well, let's say, applications of this model. One is actually part of why I was a bit surprised, is that if you, at the time at least, which was 3 years ago, when we compared the performance of this, and this was, I think Leo is here around somewhere. It was an important part of this work when the model was tried and compared on a database of street scenes in Boston, in Cambridge actually, labeled by a number of undergraduate MIT students. When we compared the performance in labeling all objects in images of this type, we found that the performance, as I said, at least at the time, was very good. It was at the level of the best algorithms that existing and coming from, so to speak, engineering, from computer vision. And so I was surprised because we really did not develop this model with the idea of providing good performance in recognition tests, in natural images. The model was simply, well, I'm not whether it's simply or not, but trying to reproduce the behavior of neurons at various stages of the visual pathway. So that surprised, the fact that it worked relatively well, mimicking psychophysics, humans' performance, and working pretty well on images, was a surprise, an interesting one. And since then, we have used a similar model, quite similar. In interest of time, I will not describe it, but it can be thought as the equivalent model for the so-called dorsal stream, which is unlike the ventral stream, which is more involved with shape and the question of what. The dorsal stream is thought to be involved more with the question of where and the question of motion. And so in a similar model for the dorsal stream, we looked at the problem of classifying actions. And there is work going on in collaboration with David Scheinberg, a physiologist at Brown, on this. But in the meantime, this model has been used by Tomaso and Wei-Yan Jung to try to provide an automatic phenotyping of the behavior of mice in a cage. Now, this is becoming important because there are thousands of mice mutants that are models of different diseases, in particular mental diseases, and we are coming at the point in which we have a very good genomics. So these mice, the genotype is well defined in a very quantitative way, but the phenotype is qualitative, or almost so, because it's based on people looking at the behavior of the mice and deciding which, in a qualitative way, what the behavior is, or at most trying to label videotapes of the mouse in the cage. So we have, as an initial proof of concept, we have a system that is trained. So is this model, dorsal stream type model, that is trained at the level of the classifier to classify 8 different types of micro behaviors over action motions that take place over a few seconds, and so after training with quite a few hours of video, we have a system that you can see here the automatic labeling of what's happening here, and the system itself is at the level of human technicians, in the sense that this is the error estimated between pairs of human observers, or between this system and a human observer. And so you have there a system that can work, analyzing many hours or in real time what's going on in the cage. And we have code using GPUs, and so we can analyze in real time more than one cage. And we are then we are collaborating with several labs in the institute, or at the Broad Institute, the genomics one nearby, to characterize the behavior of various types of mutant mice and pharmacological agents. So that's kind of an application which is a little bit ironic that a system that was piled from the brain is helping biologists, hopefully making progress in understanding these mental diseases. I want to mention very briefly a couple of other things. One is about attention, and this starts from the realization, we are certainly not the first to observe that, is that if you have more than one object or you have more and more background, the performance of hierarchical feed-forward systems like the one I described tend to degrade. For instance, in what I shown you earlier, the worst performance, and the same is true for humans, but is for images which are labeled like far body there, which are the ones where the objects are small and there is a lot of clutter. So clutter is a problem for circuits, for networks of this type, and one obvious solution is to use attention as a process that focuses, concentrates processing to locations in the images and suppresses the clutter nearby. And there are many caveats in what I said. It's more difficult than I make it seem, because you have to decide in a non-trivial way where to look. But that's the basic idea, and we have recently, we have recently worked on this model, essentially a graphical model, a Bayesian model of the areas that neuroscientists think are involved in attention, like the frontal eye field, the lateral interparietal cortex, prefrontal, V4, and this model seems to help recognition and also to predict quite well eye movements of people looking at an image, either in a bottom-up way without attention or without any task, just looking at the image, or in tasks in which people have, for instance, to look for cars or for pedestrians, in which case, in the same image, the pattern of eye movements is different, so it's task-dependent. And this model does a reasonable job of predicting what humans do under these different tasks. I'll speak about it more tomorrow at the Weizmann Institute, so this is a little bit of an advertisement for the talk tomorrow. And here's an advertisement for the other talk at CNC, and it's trying to ask the question of why these models, these simple kind of stupid feed-forward models, work as well as they do. I mean, they don't work as well as our human visual system does, but they work better than I would have thought. And why? And it's a funny situation to be. We have a model, and I've been playing with it for years, and I still don't understand what it does. And I hope this will not be the general situation for computational neuroscience. We can have great models of the brain, and we still don't understand how they work, right? So the conclusion is that if you really want to understand, we should try to get some theories, not just models where there are too many parameters, but some theorems that tell you what that will not work or what may work, and what is perhaps what is the optimal for some of the parameters. So this is something we are trying to do with a number of mathematicians, in particular Steve Smale, who is now in Hong Kong. I've been working with him trying to abstract from this hierarchical organization of cortex and study the properties of hierarchical layered architectures. We don't have too many results so far, but if we have some results that should apply to a number of architectures with many layers, such as, for instance, the deep architectures of Geoffrey Hinton and of LeCun. So if you are interested, I'll try to tell you something, mostly how difficult this is on first. Here are several of the people who have done the work, and Lior is here, Ilan Lampris is Weizmann, and there are a number of other people here who did various pieces in collaboration or on the side of, for instance, the physiology. So if I have, it's already too late, but if I can stop here or tell you for 5 minutes of an initiative that we are starting at MIT, and in some sense it may be relate or relatable to IC&C. And so let me switch gears. This is an initiative that, for a reason, a number of random reasons, came to be not long ago. We are trying to spark a research initiative, a little bit like the energy initiative at MIT, which has been quite recent and quite successful, which is across different departments and schools. I say we because this is mainly Josh Tenenbaum, who's in my department, and me, with the help of Mirgan Kassour, who is the chairman of our department, and Mark Kastner, who is a physicist, the dean of science. And Shimon Ullman was also involved in some of these steps. So the idea is to get together the faculty at MIT who are interested in the problem of intelligence. And of course, you know, intelligence, we all know what it is. Although probably the best definition is this one, is a little bit like the definition of pornography. When you know what it is when you see it. But the most important thing is that intelligence in principle can be at the level of an individual mind, an individual computer, but it can be also at the level of a group, like collective intelligence, and it can be at the level of perhaps simple ensemble of molecules or so. Anyway, the fact is that over the last few years, scientists, for the first time, have the tools ranging from computers to molecular biology, to the ability of collect huge amount of data. So for the first time in our history, after centuries in which the philosophers have spoken about the problem of intelligence, but now we have the ability to ask this as a scientific question. You can ask big questions, ambitious questions, like, you know, what is really the software of the mind? We can ask certainly the question of what is the hardware of the brain, but the question of software of the mind, the question of how intelligence emerges in an infant over the course of a few months, what is the interplay of genes and learning from experience, and more importantly, how is the priors in the genes, how are they expressed? They're certainly not precise instructions of behavior. And so these are a lot of ambitious questions, the fact is that there are a lot of different faculty, as we found out with Josh, in many different parts of MIT, ranging from electrical engineering to economics, Sloan School, linguistics, which were very interested in this problem of intelligence. Of course, as a first stage, the main pillar of this enterprise should be constituted by neuroscience, which studies the brain, cognitive science, which studies the mind, and computer science, which studies how to make computers more intelligent. But as I said, there are a lot of other contributions. For instance, the question of collective intelligence is more a question for Sloan School, how organization can be intelligent or stupid, as the financial crisis revealed. Can we do something about it? What is the trade-off of communication and individual intelligence in a group of people to be more intelligent than another group? And then another pillar of this enterprise is the kind of physics and chemistry of intelligence. So, you can ask what is new with respect to artificial intelligence. And of course, there are a lot of new tools that we have, from more powerful computers to much better genetics tools, and so on and so forth. But my personal cynical comment is that there is not much new with respect to classical AI, but it is a very ambitious, very important goal, and it's time to try again. And in a sense, if you think about it, AI started in the 50s or 60s, and then, as it happens with epidemics, good ideas are infectious, and as it happens with, say, the epidemics of flu, there is a recurrence every 25 years for obvious reasons. And so about 25 years after artificial intelligence came out, there was an epidemic called the neural networks in the 80s. And now it's about 25 years later, so it's time to get infected again. And we are betting on this for starting this initiative. We got, that's the usual blurb, why MIT, and MIT had cognitive science was born there, AI was born there, maybe, and so on. And the most important thing is that a lot of people are interested in this. They think it's, first of all, a great intellectual question. It's probably the greatest problem in science. What is intelligence? What is the mind? And it can be very important for the economy, for improving as a society the way we take decisions, and so on and so forth. And there are a lot of people at the MIT faculty who are interested. You can look at some of the stuff at this website that I just set up. And we had a workshop with about 60 faculty trying to get together, and we are going to be able to fund a few initial small seed projects. The administration is quite interested to push something like this, which will cut across all schools and departments in the university. So that's a little bit of advertisement for this, but I think there are obvious links that we should explore with ICNC. Thank you very much.
questions. Any questions?
Yeah, you were mentioning in your model that there were some pictures in the rapid presentation that the model couldn't tell. And it was the same with the humans.
Mostly, not always. But,
yeah. And I was wondering, what is the intuition to get, why is the failure with these specific pictures? And I presume that in the long display, you can tell what these images are.
Yeah.
So that obviously, the long display activates the feedback loop. So again, what is the importance of the input of the feedback loop?
So the first question is really one of these questions that is embarrassing not to be able to answer. Despite having the model, we still don't know, what are the basic features, what they are doing. I cannot predict which image will be difficult for the model. Unless I say, okay, that's difficult for me, then probably that's not, I don't understand why. Yeah, and the other one, yes, in what I just mentioned about our working with attention, which would be feedback, probably not all the feedback which is going on, not feedback across the ventral stream, but feedback through parietal cortex. Attention improves recognition. But still, even with that, we don't have performance that is at the level of humans when you present those images for a long time, because then a human is basically 100%. Right? It may miss 1 in 200, maybe, because so I guess attention and feedback, those feedbacks involved in attention, play a role that helps, but we still don't know how to have a system that is as incredibly good as we humans are.
Is there a natural way to combine that with a feed-forward networks? Isn't there some extension to the model that enables to use the other direction?
So you mean the attentional feedback that we used is on top of the feed-forward model. So they are both used and play a role. Now, I think there are other, but we have not tried to model the feedbacks that are within the ventral stream. There is a recent version of a system by Shimon Ullman, who is essentially a probabilistic Bayesian type graphical model that has a bottom-up step and a top-down one within the same layer of units. And where performance in the top-down step is important for segmentation, so for localization, segmentation, but will not improve recognition. We have not tried to model these feedbacks, and I'm not sure exactly how to do it in our framework, apart from, trying to get inspiration from models like the one of Shimon.
You rightly characterize the model as relatively very faithful to the biology of visual cortex. Nevertheless, it's a huge model with many parameters.
Yep.
And I wonder, how do you balance in such a project between computational constraints and the real biology? Because obviously the numbers of neurons in each level, the synaptic strength, and so on, all these are just picked at random, or?
Well, yeah, it is a difficult balance. Also, one reason why we did not try too hard doing a really biophysically faithful model with spiking neurons and synapses and so on is partly because I have the feeling that there are too many choices already in terms of parameters, and it would be very difficult to get anything right. We're already probably beyond that threshold. Yes, we had to take the main type of data that we have and have an educated guess about what was reasonable and what was not. And I'm sure that the model is wrong. You know, models are by definition simplification of reality, and therefore they are wrong. But this project started really because I wanted this was started many years ago, because I wanted to check whether this simplistic, feed-forward, Hubel and Wiesel type of model, whether this could be rejected. So my default hypothesis were it will work so badly that we can forget about it and go on with other things. And then it turned out, in fact, it did work better than I thought it could. And so we have continued to explore it. But, you know, at the same time, it's this difficult balance in computational neuroscience between guessing some of the things that you don't know because there are no data, and not guessing too much because otherwise you are completely wrong. I don't mind being wrong if what comes out is useful for the experiment. So the way I think about this model and the way we've been trying to use it in our collaborations with physiologists is more a guide, is more an hypothesis to disprove. You know, it tells you experiments that you can do. It tells you what the answer may be, and it helps you think about the problem. But you know, there are many assumptions in the model which are mainly shared by physiologists right now. Something like there are labeled lines, right? A neuron essentially does not change its properties after this unsupervised learning. They may turn out to be wrong. And so, anyway.
So, it's becoming very popular, as far actually we had a talk like this just last week here from Mr. Ball, to hypothesize is actually it's going the other way around. I mean, here in Jerusalem, we must mention the reverse hierarchy model or things like this. Essentially, we first guess what we see and then we actually verify. And it's something which I personally like very much, because hypothesis testing is a lot more efficient than generative reconstruction of the whole image, bottom up. So in some sense, it seems that you're still using this view that we look only at snapshots of reality, one at a time, but in fact, we have time that allows us to make predictions, and very coarse prediction, and then verify them. So I just wonder if you see this as part of the truth, or
yeah, well, the reverse hierarchy, I like the idea very much, but you know, this is, as I said, is kind to make a quantitative simple models that, you know, starting with Hubel and Wiesel, and you will have to explain anyway that we can recognize objects in a flash, and there are other things one has to account for. For instance, that it takes, you know, I did not mention this, but in these experiments on IT, and confirming earlier data, you find that the delay is around 80 to 100 milliseconds in the activity of IT. And starting with the first 5 or 10 milliseconds of spikes, you have already enough information to read out from this activity. So these are all things, and this amount of time is quite short if you have to do a lot of feedbacks or iterations between, say, IT and V1. So I personally think that the back projection are certainly very important for normal vision. But I'm not sure whether they play a role in this fast kind of rapid categorization. Of course, it's quite possible that in addition to the ventral pathway, there may be extra cortical pathways involved in some aspects of it. It would be a bit difficult to believe, and it's not impossible, that it's all done outside the visual cortex or the ventral stream. We'll see.
Let me ask you the last question. So my take from the model is mostly what you're telling us, that there is this model that came from neuroscience and turned out to work very well in engineering applications and has been used for neuroscience, but has it had the same effect on neuroscientists in terms of changing their understanding of the brain, modeling the brain?
Well, I don't. I think some people. Let's see. It's a difficult question. First of all, it is always difficult to convince neuroscientists working with monkeys to do experiments for you. And it has been a little bit easier with this model, but still not easy. And the reason mainly is that, as one of the physiologists told me, I have in my life about 20 experiments. No more. Because each one takes like 2 years. Sorry?
Expecting a long range of life.
Exactly, that's an optimistic view. And so I'm just saying these experiments are very expensive, right? So the model was not received in such a way that now everybody wants to do experiments related to it. Certainly not. But on the other hand, I think for many physiologists, seems to be that models of this type are what they expect, so they're not too surprised. I think the question is going beyond them. Today, and with reasons, there is a lot of interest in attention and oscillations and related phenomena. And so I think everybody wants to go beyond purely feed-forward, which I think it's right. I'm not sure I answered your question, but...
How about going to rodents? More than 20 experiments.
Well, people are starting to go to
but
rodents.
Is there any use of such a model of vision in rodents?
Probably yes. I mean, the problem with rats and mice, and some people are actually starting big real efforts there, is that very little is known about, relatively little is known about the anatomy of visual cortex beyond V1. So even knowing where to put your electrodes is a problem. But people, as I said, are trying to do it. And the behavioral part, which is the other part, training a monkey is difficult, but it works for relatively difficult tasks. For rodents, it's more difficult. But some people have had success recently in training rodents for doing recognition. So, yes, I think this is coming.